Hello, everyone. My name is Mite Mijalkov, and I work as a postdoctoral researcher in Karolinska Institute. And first of all, let me say a big thank you to the organizers of this conference for giving me this incredible opportunity to be here and present my research into sex differences in the functional brain connectivity throughout aging, and present to you the novel method of multilayer connectivity that captures these differences better than the conventional methods. So over the last decades, there has been an increase in the human lifespan. However, unfortunately, this has not been matched by a considerable increase in the health span. And in particular, aging remained a major risk factor for several disorders. However, it has been shown that not all individuals age at the same rate. And in particular, some of this intersubject variability is partly due to sex. So it's important, therefore, to study these sex differences throughout normal aging, because it could explain why men and women show different prevalences of several diseases, but it also could provide a benchmark against which the disease-related studies could compare their findings. And also, it could fundamentally change the field and how one treats statistically age and sex in their studies involving functional connectivity. However, while a lot of studies have been dedicated to assess these changes throughout aging, with respect to sex differences, these changes in functional connectivity has been understudied. And this is part due to the availability of only small samples and therefore low statistical power that is available for these studies. And therefore, here in this study, we set out in a large cohort based on UK Biobank to show that the novel method of multilayer connectivity can capture these sex differences in functional brain connectivity throughout aging. And this will be the method that I will present to you during the rest of this presentation. So starting with the cohort, the data that we assessed was pre-processed data from the UK Biobank researchers, and it involved resting state functional MRI data in which the whole brain functional connectivity for each participant was summarized by a 21 by 21 connectivity matrix. In this matrix, the nodes represented the several resting state functional networks in the brain, and the connections between them were derived by a person's correlation. An example of this network you can see here on the left, where the positive correlations are shown in red, while the negative correlations are shown in blue. And this data was available for a large number of women and men that were in the age range between 47 and 79 years of age. So moving on to the method that we used to analyze this data, we first, starting from the matrix that we obtained from the UK Biobank, what we first did is that we split the networks of positive and negative connections, and we analyzed them separately. So the topology of these networks was analyzed by clustering coefficient, which basically quantifies the degree of functional segregation in the network, but also we calculated the global efficiency, which basically quantifies the levels with which the two different brain regions can communicate or interact with each other. So as I said before, one big disadvantage of this single layer analysis is that we analyze the two networks separately from each other. So therefore, we don't have a means to assess their interaction. To allow for this, we first combined these two networks as two separate layers in a single multiplex network. And in this construction, each node in layer 1 was connected with the corresponding node in layer 2. To assess the topology of this network, we evaluated two previously described coefficients. The first one is the participation coefficient, which tells us how similar the connectivity patterns are in both layers. But also we evaluated the clustering coefficient, which is just an extension of the single layer clustering coefficient. But in this case, it considers the triangles that consider edges that are present in the two layers, as you can see here in the figure. But again, this approach, while good, it has its disadvantages. And one of the big disadvantages is that the interaction between the two layers here is fixed and it's unitary. So in order to assess this disadvantage, we propose that a multi-layer analysis would be more sensitive to these differences. And in this type of analysis, we allow for a node in one layer to be connected with all nodes in the different layer. Moreover, the interlayer connections can have their own weight. And in our analysis, we treat this weight as a free parameter, and we evaluate the topology of these networks as a function of these weights. However, as in our analysis show that the topology is stable across a wide range of these interlayer weights, in the rest of the presentations, I will just show you for one particular weight that showed the biggest differences. So because this type of approach is novel, we also came up with two novel measures to evaluate the topology of this type of networks. So to evaluate the global efficiency of this network, I will use a toy model to explain you how to calculate that. And consider here, as shown in the figure, that one node in layer 1 is connected with all the nodes in layer 2. So then one can evaluate the same layer global efficiency, which is basically the global efficiency of the network consisted of the nodes that are in the same layer of node 1. Then we can evaluate the opposite layer global efficiency, which is basically the network consisting of node 1 in layer 1 and all of the nodes in the opposite layer. And then the global efficiency of node 1 will be given by the subtraction of these two measures. Once we calculate the average of this measure over all of the nodes in the given layer, we will obtain the global efficiency for the complete multi-layer network. And analogously, one can calculate multi-layer clustering coefficient by calculating same layer and opposite layer clustering coefficients and taking their subtraction. So moving on to the final part of my presentation and the results that we obtain. So what you see here is the corresponding measure plotted on the y-axis as a function of age of the participants. And the areas here show the baseline fit and the confidence intervals of the linear models that have been fitted when the given measure was used as a dependent variable, and age squared, sex, and their interactions were used as a predictors. So what this result shows us is that in the single layer network topology, men tend to have a higher global efficiency in both positive and negative networks. However, these results also show that these differences are expected to be constant throughout age, as this did not show any significant age or age squared interaction with sex. On the other hand, in the functional segregation properties, we didn't find any differences between men and women, which you can see by the stars here that show the between group differences, whether they're significant or not when calculated by the permutation tests. So moving on to the multiplex network topology, we see the first difference from the single layer topology, in a sense that while all measures show that multiplex participation, multilayer clustering, and multilayer global efficiency are higher in men when compared to women, they actually show that this difference is more pronounced for younger adults. However, this difference then dissipates as people get older, to the point that after 75 years of age, there are no discernible sex differences in the functional connectivity between men and women. And this can be shown by the significant age-sex interaction in the linear model. Now, to evaluate the clinical relevance of these results, we run a partially squared analysis where the dependent variable were several cognitive, cardiovascular, and structural measures that were differing between men and women in our sample. And what this analysis shows is that, as you can see by the red squares with which I denote the multiplex and multilayer measures, in all of these measures that differ between men and women, multilayer and multiplex measures appear as a significant predictors of the variance in these measures as a function of age. And finally, we ran a GWAS analysis of all of these multi-layer measures, and we found out that these measures are associated with several genes that has been shown to be implicated in several physiological processes that change with aging, which again confirms that multi-layer connectivity has certain genetic influences on it. So with this, to summarize, I hope that in these short presentations, I have convinced you that multi-layer connectivity as a novel method is an important method that can capture the sex differences in functional brain connectivity throughout aging much better than the standard conventionally single layer measures. And we have shown this in a large cohort, which consisted of around 37,500 UK Biobank participants. And finally, I would like to thank to all of the people who have been part of this work, and especially since all of these results were derived with BRAF 2.0, I would like to extend special thanks to the people who helped and developed this software, and they present this software in a poster at this conference. With this, I would like to thank you for your attention, and please feel free to ask many questions during the in-person conference or email me any questions you might have.